Okay, so Brenda already talked about this part, but I wanted to go into a little bit more detail with the 15N and the 14N. So we know that there was nitrogen that was 15, so using different radioisotopes we can tell which generation was used. And so then basically after this entire experiment was performed, you would be able to tell if the DNA was from a specific generation, if one was from the parental and one was from the daughter. So then you could tell, once you put the DNA strands together, if both strands are from the parents or from the daughters, or if one of them is daughter, one of them is parent, or if they're dispersive, like we talked about. So you could tell semi-conservative, conservative, or dispersive. Okay, so we had 3 projects to do about facilitating such things. So the first part of that was examining the structure of the alien DNA that we were given, and we had to explain the similarities with the eukaryotic life you could find on Earth. So here on the right, you can see the original alien genetic material. We know it's double-stranded, and that the nucleotides bond via hydrogen bonds, shown here as these dashes, and the pyrimines bond with the pyrimidines for uniform width, and they used adenine, which is kind of the only nucleotide in common, which we can go into more detail right now. Second question. We could examine the structure of the alien DNA again, but this time we explain the differences with the eukaryotic life you could find on Earth. So here's the same picture, but now we can look that it's parallel. And if you look at regular DNA in eukaryotic cells, it's anti-parallel. And that's very important because if it's just parallel, then the specific groups are going to repel each other and you can't keep a uniform structure in the same way. Now, a big difference is that it uses different nucleotides, like it uses isoadenine, isothymine, isocytosine, and it uses beta guanine. And it sort of seems unclear here, but it's supposed to say it uses iso for all of these things here. And of course it also uses uracil, and that's only in eukaryotic RNA, not the DNA. So it's a big difference. Finally, we can look at our last question, which both Brenda and I facilitated on. This question called for us to compare and contrast the process of replication between the alien sample and eukaryotic life found on Earth. So in the comparison, we can see that they both contain material from the parental and the daughter strands. And the thing is, if you look at regular eukaryotic DNA, you'll have one strand, and that'll be the parent. And if you look at the other strand, that'll be the daughter. So this one's pretty different. If you look closely, you can see that there are italicized and regular font. And the italicized are supposed to be the new DNA that is formed after the replication, and the regular font, it would be the parental. So parental, daughter, you can see they're both different kinds on the same strand, and that's dispersive replication like we went over, but eukaryotic is semi-conservative, so that's another big difference if you're comparing the different kinds of replication. But this also helps us out with the experiment we talked about before. We would expect it to be dispersive. Oh, go back. Or don't go back. Okay. Pretty much what that was supposed to say was that we were given a piece of DNA to examine, and it had, I believe it was 88 nucleotides of adenine that we know about. So this picture over here that I just shown, that was just to show you that, we know that adenine is going to bind with thymine, and cytosine is going to bind with guanine, and these are just the nitrogenous bases that we use. So, first question is how many nucleotides in the piece of DNA can contain the base guanine? So we already know from before, I'm sorry, I skipped ahead, that there were 410 nucleotides total, minus the 88 adenine nucleotides. But we also have to subtract 88 thymine nucleotides, because assuming that there aren't any mutations, we know adenine will only bond with thymine, and being that there are 88, there must also be 88 thymine. So 410 minus 88 gives us 234 nucleotides left. 234 divided by the two complementary bases of cytosine and guanine, because you can't just have a random amount of guanine and expect the cytosine to just hang out there. They have to bond together, which means you take 234 and you divide it by 2, so you'll have an even amount of guanine and cytosine nucleotides. This means that the answer to our question is 117 guanine nucleotides. Second question. How many phosphate groups would be found in the piece of DNA? There are 410 nucleotides, and there are 408 phosphate groups, and 3 phosphates at the 3' end on one DNA strand, and 3 phosphates at the 3' end on the other DNA strand. Sounds familiar? It should. This is what Brenda was talking about in part 1. This is the way I explain it, as she was talking about. We have different ways of explaining it. So if you know that there are 410 nucleotides total, you might first assume, oh, okay, there's a phosphate group for each nucleotide. You can even look at this picture right here. But then you have to think about it again. If you look at the 3' end, it's going to have 3 phosphates attached to it, because when it's in the middle, that's when the phosphates break apart their covalent bonds, and then you're left with a pyrophosphate that's separated from the molecule, and then the regular phosphate, which covalently bonds the actual sugar bases together to form the sugar phosphate backbone. So now you know you have to subtract 2 of those nucleotides from the beginning, so you know you already have 408 phosphate groups. Then you can go back to those 2 and say, of that one that has 3 phosphates at a 3' end on that DNA strand, and the other strand, which would of course also have a 3' end, will also have those 3 phosphates. Just, you know, you have to add them. So 408 plus 3 plus 3. So that pretty much gives us our answer. And if you didn't add it up, you know, it's 414? Yeah. This is just a close-up of the picture I was talking about. And that's that phosphoester bond, and there's the pyrophosphate there. Third question. How many molecules of uracil will be found in a piece of DNA? So we know there are 88 nucleotides of adenine, and in the case of RNA, adenine would pair with its corresponding 88 nucleotides of uracil. So you might say 88 at first. But this is a strand of DNA, and DNA doesn't have uracil in it. So the answer is 0. And, you know, this just goes back to show you, yes, A with T, C with G, no U. In the DNA molecule, which molecules are not bonded together? So here we were given a list, A, B, C, D, and E. A has adenine and thymine, and we looked at the picture, we know they bond together. Deoxyribose and phosphate groups, those bond together too, with the sugar phosphate backbone. Cytosine and guanine, they bond, and the deoxyribose and adenine, well really it's deoxyribose and any of the other nucleotide bases, because the ribose is attached to that. But if you look at E, the phosphate groups in the cytosine are not attached. The phosphate is what's holding those sugars together, but it doesn't touch the actual cytosine, guanine, adenine, thymine, none of those. Those phosphate groups are bonded to the ribose sugars and not the nucleotides. Now you can see this thing again in case you really wanted to see it before. And then we can go into the 5th question, which was asking us, out of these 4 options, I'm sorry the top's cut off, I'll show it later, but out of these 4 options, which one most closely resembles eukaryotic replication? So here, you can be given an original strand, that we can refer back to later, and shows you just basically, there are no mutations. And you can look at conservative. I'm sorry, this is cut off too. This is 5, 3, 5, 3. And you know it's conservative because this has the parent strand. Remember, regular font is parents. And then this is the daughter strands put together. And we know that because they're italicized. So this is conservative, and that's not how eukaryotic replication is. Then look at dispersive. We talked about this already. You can see italicized and regular font are put together on both strands. Then there's parallel. I mean, look at this. 3 does not bond with 3, and 5 does not bond by 5 going this way. This is supposed to be 5 to 3, and then this is 5, and that one would be 3. So you know this is parallel. This can't be right. Even though it's still, semi-conservative. Which is this. This is option D, and this is the answer. It has to be semi-conservative, it has to be anti-parallel, and it most closely resembles the eukaryotic DNA for this reason. And that is the end of our Prezi.